I'll be sharing some of the insights I've gained from sequencing the RNAs of microbes native to skin, and I'd like to thank the organizers of MVIF for the chance to speak. Now, the skin surface consists of diverse microenvironments with distinct pH, temperature, moisture, and sebum content. Now, these niche-specific physiological differences influence resident skin microbes. For example, we know that oily surfaces like the forehead and the scalp support lipophilic bacteria and fungi, and that differs from the dry, low biomass sites like the forearm. Understanding the role of members in the skin microbiome is crucial because we know that they influence many skin phenotypes such as body odor, itching, and even eczema flares, for example. Now, additionally, while the human genome is estimated to contain about 20,000 protein-coding genes, there's an estimated about 10.9 million non-redundant genes across skin microbiomes of different people, and a substantial fraction of them have unknown function. So, as scientists, we are keen on using different methods to study the functional role of our tiny neighbors, to not only understand who is there, but what they are doing on our skin. And there are many complementary tools to study microbiome functions, such as sequencing-based approaches. You can culture microbial isolates and assemble the genomes and analyze the gene content. Then we can test for different host microbial interactions through various animal, tissue, or cell models, as we've seen from our lovely keynote speaker just now as well. But, to date, most published microbiome studies, especially in lower biomass tissues such as the skin, rely very heavily on different forms of DNA sequencing, for example, 16S or shotgun metagenomics to profile microbial communities. Now, this is a powerful approach for discovering which species and genes are present, but it is also limited because it cannot distinguish between living or dead species. It also cannot tell us which microbial genes or pathways are expressed in vivo. So this led us to pioneer efforts to measure in vivo microbial activity on skin using RNA sequencing. Our team optimized a skin microbial sampling protocol where we tested various experimental parameters such as swabs or skin tapes, the isolation methods, the extraction methods. This is in order to obtain sufficient quantities of material, and we also had to deplete the ribosomal RNAs from human, bacterial, and skin fungi using custom oligos. We tested, of course, the robustness of our approach first in mock communities, and we found very high reproducibility between technical replicates at both the species and the pathway level. So this gave us some confidence in our approach. We also had to then combine our experimental protocol with a computational pipeline that is customized for analyzing the skin metagenome and metatranscriptome, using a more comprehensive gene catalog that's customized for skin with both Asian and Western representation. This workflow allows us to track which species are active on skin, which microbial genes are expressed on skin, and what their possible functions are. And this is all hosted on GitHub, so you can actually use this pipeline for yourself as well. We applied this approach to a cohort of healthy individuals across 5 different skin sites, representing different microenvironments, oily, moist to dry. And we obtained several million paired microbial reads even after rRNA removal and deduplication. And this work has been published in Nature Biotech. Look at the full story if you're interested in the details. But I'll be focusing on a few interesting observations. Now, the first interesting observation we made when comparing paired skin metagenomic and metatranscriptomic data was the striking differences between the two measurements. Now, here I'm showing data from scalp, and we sorted this data in order of C. acnes metagenomic abundances. So you can see the top row here in green. So at the DNA level, in most people, most of the reads belong to a common bacteria called Cutibacterium acnes. So not surprising. But what's surprising was that these are not the most active species at the RNA level. In fact, if you look at the bottom row here, at the matching RNA metatranscriptomes, we found that Staphylococcus and Malassezia, which is a skin fungi, contribute disproportionately to metatranscriptomes than would be expected from their metagenomes. In this example, I've highlighted 2 particular individuals who, in an abundance plot, where their metagenomes are dominated by C. acnes in green, but the metatranscriptomes are actually heavily represented by Malassezia fungi and other microbes. Now, these differences are, of course, because metagenomes reflect genome copy numbers, whereas metatranscriptomes measure the active biomass. So you kind of need both to have a holistic understanding of what your skin microbes are doing. We also see the same trends across different skin sites, not only in scalp, with Malassezia and Staphylococcus being notable sources of active transcription, despite their relatively low DNA abundances. And this, again, is information that you would not normally get from metagenomics alone. So metatranscriptomes, we think, are a valuable data set when you combine this with differential expression analysis to look for signatures of microbial adaptation to different skin microenvironments. And notably, I think we can even find gene expression differences even between sites that seem to be superficially similar. For example, the cheeks and the scalp are known to be lipid-rich. But actually, if you compare skin fungal gene expression between the two sites, we found that ether lipid and glycerol phospholipid metabolism was relatively upregulated in cheeks, while peroxisomal function and some carbohydrate metabolism was relatively upregulated in scalp. Now, we believe that some of these observations can be explained due to variations in vivo nutrient availability between these two sebaceous sites. For example, scalp is known to be richer in free fatty acids compared to facial skin, and the oxidation of fatty acids is, of course, mediated via the peroxisome in these fungi. In contrast, glycerides are more abundant on facial skin than on scalp, and these could be then broken down by enzymes involved in ether lipid or glycerophospholipid metabolism. So in my previous slide, I showed that Malassezia fungi actually secrete phospholipase C to break down some of these lipid classes as well. Then another useful approach of metatranscriptomics on skin is to measure in vivo antimicrobial gene expression. This is important because there are many good reasons to minimize the use of topical antibiotics to treat skin infections or diseases, because we don't want to spread antibiotic resistance and we don't want to unintentionally kill our beneficial skin commensals. So instead, we can examine ways in which skin microbes perform interspecies warfare. And it was reported a couple of years ago that some C. acnes strains on skin contain a gene cluster encoding a thiopeptide bacteriocin known as cutimycin, which can inhibit the growth of Staphylococcus epidermidis within the same niche. Conversely, we know that some Staphylococcus species and strains can also inhibit the growth of C. acnes by secreting peptides known as phenosoluble modulins. Now, using metatranscriptomics, we now have the opportunity to measure these interactions in vivo. So we split our samples into those with detectable thiopeptide expression at the RNA level in C. acnes and from those without. We then looked at the metagenomes, so on the y-axis, and found a higher ratio of C. acnes to different Staphylococcus species at a DNA level. And this is particularly true at sebaceous sites where C. acnes is much more active. And this suggests inhibition and competition again. We did the same for RNAs involved in auto-inducing peptide and phenosoluble modulin expression in Staphylococcus, and we saw the reverse effect, right, where there's a lower ratio of C. acnes to Staphylococcus. What's even more interesting is that we also saw that other uncharacterized thiopeptides with potential bacteriocin activity were expressed by C. acnes. Now, many of these peptides have a very different amino acid, primary amino acid identity to cutimycin, and they're only predicted to exist computationally. But now we have more evidence that they can be expressed in vivo. So we think that metatranscriptomics is an exciting approach to discover more interactions between microbes. So in summary, I hope I've convinced you that we've developed an optimized skin RNA-seq protocol that's robust across different skin sites and can be deployed in the clinic using just skin swabs. It's important to note that RNA is different from DNA abundances, so activity is not the same as organismal abundances. And we can then use RNA-seq to now measure signatures of microbial adaptation to their metabolic requirements and also measure key antimicrobial functions and interactions and to discover new ones as well. So what's next? And because we of course want to understand disease, so we are hopeful they can deploy this approach and collaborate with people to in disease cohorts. So you can imagine that you can apply RNA-seq, DNA-seq, and other omic layers to a disease cohort before and after disease to discover host and microbial gene and immune pathways that are associated with disease. And if you combine that with microbial strains, you now have a much bigger understanding of what contributes to disease mechanisms. And you can train a model then perhaps to predict the propensity to get some disease like eczema flares that can recur. And then, of course, you then validate these in a validation cohort and hopefully get a set of validated clinical markers, right? So this work is only possible through the guidance of my mentor Niranjan from Genome Institute of Singapore and my team members and various collaborators who are highlighted in bold. I'd like to thank my funding agencies as well, for the Asian Skin Microbiome Program and the Singapore and various Singapore institutes, Skin Research Labs and the Genome Institute of Singapore. And also, again, I'd like to thank the organizers and for inviting me to speak, and the audience for your time and patience. Now, thank you, everyone, and happy to take questions.